assume that you're seeing my screen, right? Oops, something weird happened. This one, okay. Okay, so thanks for the invitation. So what I'm presenting here is basically an update to a phylogeny that we just published in Cladistics. I think it was this year or the year before. Basically, what we have done here is to incorporate more taxa, more characters, and perform different analysis. So as you can see from the title, it's a phylogeny on the complex thyloliverworts. Now, what are the complex thyloliverworts? These are a group of thyloid bryophytes, which comprise a bit less than 400 species. Let me move this over here. And which are characterized by having this gametophyte, which is the main body of the plant that you're observing here, which in the flowering plants are reduced to a mass of cells within the flower. In this case, the gametophyte is the dominant phase of the life cycle, and it's characterized by being dorsiventral in its symmetry, that is to say it's compressed. And internally, it may develop different tissues that perform or conduct different functions, such as being photosynthetic, for instance. And another feature of interest of this group, which is actually a synapomorphy, is the presence of polarized spores. That is to say, spores that differentiate a distal phase from a proximal phase. The proximal phase being the phase of the spore, which is in close contact with others during the meiosis. Systematically speaking, the classification scheme for this group is based almost entirely on molecular phylogenies. Something curious about the phylogenies of this group is that throughout time, authors have attempted to increase the number of molecular markers, but in doing so, they started to exclude morphology from their analysis. So a consequence of this is that the current classification scheme, which is based, as I already said, in mostly on molecular phylogenies, has no synapomorphism for most of the clades. And in addition, clades within Mercantiales have remained weakly supported despite increasing amounts of molecular data. Because of this, in 2017, we performed the second combined analysis for this group, which was actually the largest in terms of data volume. And we analyzed over 100 morphological characters, 11 molecular markers, which have been analyzed before in a paper by Villarreal et al. And also we analyzed 56 extant taxa. And among other things, in this study, we were able to find synapomorphisms for several clades that corresponded to others and families. And something that all these phylogenies had in common is that they excluded fossils. They never incorporated fossils in the analysis. So what about the fossils for this group? Liverwort fossils are commonly at pressions, that is to say compressions or impressions, which rarely include cellular details. And amber fossils, even though they may preserve some fine-grained details, they are more common in younger strata. So the data from older strata is basically missing from most of the phylogenies. So arguably, because of the fact that bryophyte fossils are considered too fragmentary and polypreserved, they have been rarely included in phylogenies, except for two of them. One is a paper by Alex Vipos and colleagues on a group of mosses and Polytrichaceae. And the second one is actually this one, for which I'm giving this sort of update for the complex thyloliverworts. Now, an impact of excluding fossils from analysis is related to the dating analysis. For this group, most of the, well, actually, all the dating analysis have been performed under the molecular dating approach, in which the substitution rates of different molecular markers have been employed to estimate the divergence times. And the authors, Villarreal et al., for this specific group, they employed a single calibration point, which was Mercantites cyathodoides, to constrain the most inclusive level node for their phylogeny. And from this, they inferred that Mercantiopsida radiated in early Permian and that most of the extant genera radiated during or after the Cretaceous. Now, evidently, a downside of that analysis is that they employed a single calibration point. And as we know, the more calibration points we use, the more likely is that our estimates of divergence times are accurate, or at least they are improved. And in addition, something that applies to the molecular dating approach in general is that nodes are arbitrarily constrained. And that is to say, the actual phyletic affinities of the fossils are not taken into account. So having this in mind, we decided to perform a series of studies, basically to evaluate the phyletic affinities of the fossils and their impact on the inferred relationships, if any, and to estimate the divergence times following a total evidence approach, and also to evaluate morphologically macroevolutionary trends on morphological characters. So to do so, we expanded our previous data set so that we incorporated fossils, more morphological characters, and the same 11 molecular markers analyzed by Villar et al., which published the largest molecular phylogeny for this group. So for the 8 fossils that we incorporated, 5 belong to Mercantiopsida. This is an example of some of the fossils. And 3 to Hungermaniopsida, which is the sister groups. It's another group of liverworts. For the morphological characters, we score them from the gametophyte and the sporophyte, from personal observations and literature. And as I already said, we incorporated the same molecular markers as that of Villar et al. So we first performed parsimony analysis under equal weighting, and also explored the sensitivity of in the placement of the fossils under extended implied weighting under 3 different concavities, and following 2 different approaches. On the one hand, we down-weighted individual molecular characters according to their homoplasy, and assuming that missing entries contribute to homoplasy upon being observed, and also down-weighted molecular blocks according to the average homoplasy. So from the equal weighting analysis, we observed that results were in general agreement with our previous studies based on extant taxa. And for example, morphologically simply taxa, that is to say those taxa that don't have internal tissue in the gametophyte, were more closely related to each other than to morphologically complex taxa, such as Mercantiaceae, over here, for instance. And as it was the case in molecular phylogenies, in previous molecular phylogenies, clades within Mercantiales remain weakly supported, these specific clade nodes over here. Something more important is that we recovered novel systematic affinities from our phylogenies, at least for 3 fossils, whereas all the systematic affinities that have been suggested by the authors of the remaining fossils have been supported. In the case of Palavicinites, which is a fossil in the outgroup in Hungermaniopsida, we recovered this as sister to Pelide, or a node which could correspond to the subclass Pelide, instead of being nested within this clade, which should correspond to Palaviciniales, as suggested by the authors in the original description. And as for Mercantites cyathodoides and Wallengensis, here indicated in arrows, we recovered this in different positions, a sister of to Neo-Hudsonia and Mercantia polymorpha, respectively. And the thing is that for both of these fossils, the authors prefer not to locate these fossils in specific categories below the order Mercantiales, mostly on the basis that these fossils lack diagnostic characters. So in this case, our results somehow provide the first evidence that these fossils could be placed in a specific or less inclusive taxonomic category. So regarding synapomorphisms, I'm just giving an example of a few fossils, not all of the fossils that we have incorporated. We also recovered novel synapomorphisms for some that support the position of some fossils. For instance, for the fossil Blasites, the authors argued that this fossil should be included within the family Daciaceae. and that a diagnostic character or a character that should support this position is the presence of a flask-shaped conceptacle, which is this structure that you observe here within the circle. And instead of recovering this character as a synapomorphy, which should support the position of the fossil in the family, we found different synapomorphies, such as the presence of a left thallus. Here in the fossil, you can observe here in the margin of the thallus, a double midrib, which is quite clear here in the extant blasia, and the presence of asexual gemma for asexual reproduction. The thing is that the flask-shaped conceptacles, which should be a synapomorphy for this group, or at least a diagnostic character for the placement of this fossil in the family, is reconstructed as an ambiguity between well, the flask-shaped conceptacle, and the alternative state is the presence of cavities, which are observed in the taxon Cavicularia. And something similar applies to Marchantites cyathodoides and Marchantites wallingensis. We found novel synapomorphisms for the position of both fossils. And in the case of Marchantites cyathodoides, the synapomorphy that supports its placement is the presence of smooth rhizoids with both thin and thick walls. In bryophytes, rhizoids, which are these tubes that you observe here, are an analogy to the roots in vascular plants. And these outgrowths that you observe here in the inner wall of the rhizoids are pegs. Now, in those cases where these pegs are not developed, bryophytes, or the liverworts in particular, may develop a thickened wall, but not all of the taxa necessarily. So the presence of both smooth rhizoids with different type of thickened walls and thin walls are, in this case, a synapomorphy for this specific clade. And in the case of Marchantites wallingensis, we have several synapomorphies. For instance, the presence of asexual gemma, scales, which are observed here in the ventral side of the thallus. Here you have a picture of the fossil on the left, and on the right, an example of an extant taxa, an extant Marchantia. And of course, also the presence of air pores, which again, you have here an example of an extant Marchantia and a fossil. As I already said, for both fossils, the authors prefer not to give a precise category or locate these fossils into a specific family due to the lack of putative diagnostic characters. In this case, we have found synapomorphies that support both of these positions. And finally, Rhyxiopsis, which is a morphogenus that was proposed to accommodate those fossils with rosette-shaped thallus, such as this one over here. The author suggested incorporating this morphogenus in the family Rhyxiaceae upon the observation of rosette-forming thallus. In this case, we also found that ventral scales are also a synapomorphy for the position of these fossils within the family Rhyxiaceae over here. And this is particularly important because not all of the extant Rhyxia are rosette-shaped in their thallus. So upon incorporating more fossils and more extant taxa, these synapomorphies may change over time. And this is an example of how a scale looks in the fossil. So as I already said, we also evaluated the sensitivity in the placement of the fossils under different weighting schemes. And for 3 out of the 8 fossils, we found that they had different positions depending on the weighting scheme. However, in general, we could say that topologies under extended implied weighting were quite similar to that under equal weighting, and the similarity measured on the basis of the number of SPR moves was beyond 90%, and the proportion of shared nodes was over 70%. Generally speaking, the most unstable fossil for this group, in this analysis, was Paravicinites, which showed around 6 multiple alternative positions in a single under a specific weighting scheme with a concavity of 10. And for Mercantile Cyathodoides and Wallensiensis, we observed two alternative positions throughout the different weighting schemes. And interestingly, one of those involved both fossils as sister to each other and within the family Mercantiaceae, which is interesting because both of these fossils somehow resemble an extant Mercantia, even though only one of them was recovered in the family Mercantiaceae under equal weight. Now, as I said, we were also interested in estimating the divergence times. So from this combined data set, we needed to perform Bayesian analysis. The first one was a Bayesian non-clock analysis on the complete data set. And because unstable terminals could prevent chains from converging during the dating analysis, what we did was to subject these non-clock topologies to ITER PCR as implemented in TNT. And after excluding the most unstable terminals, using a different outgroup, different routing taxon, we also evaluated the fit of 6 different models to this reduced data set. And finally, upon finding the best fitting model, we conducted the total evidence dating analysis. And I'm just reporting the results from this specific model. So the Bayesian non-clock analysis were quite in agreement with the parsimony trees under equal weighting, except for one clade, which is this clade composed by Neohudsoniales and Mercantiales, although it was recovered with low support. Again, the clades within Mercantiales remain weakly supported. And something interesting is that if we consider only the compatible groups in the post-burning topologies, the same systematic affinities between fossils and extant taxa as recovered in the parsimony analysis are supported. However, if we consider the majority rule consensus, which I guess it might be the standard practice in Bayesian inference, we observe that some fossils were quite unstable, and particularly the fossil Mercantile Cyathodoides was the most unstable in this analysis. And this instability somehow questions decisions made by previous authors who constrained the most inclusive node in their molecular dating analysis. What I mean by questions this decision is that the authors never explored different constraints in the analysis. I mean, constraining a less inclusive node, of course. And the total evidence dating analysis, as compared to the non-clock analysis, recovered 3 out of the 6 fossils that remain in the reduced dataset in different positions, usually a sister to more inclusive nodes. For instance, Metzgeriothallus sharonii, which is the oldest macro fossil for bryophytes in general, was recovered here as sister to Hungermaniopsida instead of being nested within the group. Mercantites wallensiensis, instead of being in the family Mercantiaceae, was recovered here as sister to Neohudsonia. And Rixiopsis sandalingensis, instead of being recovered within the family Rixiaceae, was sister to the family. More importantly, the divergence time estimates were older as compared with the molecular dating analysis published before. For instance, Mercantiopsida radiated in late Devonian instead of early Permian. Mercantiales diverged during early Permian instead of late Triassic, and Rixiaceae radiated in early Jurassic instead of early Cretaceous. And the same applies to the outgroup, although I'm not particularly focusing on the outgroup, except for the molecular dating analysis by Cooper et al. And we assume that the differences with the paper by Cooper is probably explained by different taxon samplings and different calibrations point that they have used in their paper. So for the last part, as I already said, we were also interested in assessing certain macroevolutionary tendencies in morphology. So for this, we employed the same morphological dataset and the topologies from the total evidence dating analysis. We were interested in assessing the character change rate for morphology, and to do so, we first performed a character optimization in TNT, and the character change rate was estimated as the number of changes relative to the branch length as implied by the dated trees, and the number of unscored characters. This is essentially, I forgot to include the citation, but this is an equation proposed by Graham Lloyd. And on the other hand, we computed the disparity for this taxa. To do so, after performing the character optimization, we computed a distance matrix, and upon this distance matrix, we we performed an ordination, and after conducting the ordination, we estimated the disparity as the median Euclidean distance from the centroid. This latter part was done in the R environment and using the CLADIS and disparity packages. So the morphology change rate was quite low throughout the phylogeny, but there were also episodes of relative high rates which alternated with these low rates. Particularly here in the branches subtending the Marcantiopsida and Marcantid radiation, the rate was quite high, and this spanned the time lapse between the Devonian and Carboniferous, and also the rate was quite high during the radiation of Oxymitriaceae and Ruxiaceae in the time lapse between the Triassic and Jurassic. And also, as you can observe from this figure, the terminal branches usually increase the change rates, which corresponded to the time lapse after the late Cretaceous in most of the cases. So the disparity, which could be considered or interpreted as a proxy to the morphological diversity, increased through time. And it does it in 3 different stages. We first observe a fast increased period up to early Mesozoic here. And throughout most of the Mesozoic, we observe a sort of plateau in which the disparity values hardly increased. And from late Mesozoic onwards, there is a slow increase in disparity. Now, as long as the disparity doesn't increase that much from this after this plateau period, we could somehow interpret this or argue that liverworts were already diverse, morphologically speaking, by the mid-Mesozoic, or at least they were so in a comparable manner to what they are today. Moreover, if we take into account the number of lineages, which is the dashed line in this plot, we observe that the pace at which the number of lineages increase is higher than that of the disparity. And we could argue from this that these, let's say, novel lineages that were appearing after late Mesozoic were not contributing considerably, at least, to the values of to the disparity. Or in other words, these novel lineages shouldn't be exploited in empty spaces or novel spaces in the morphospace. And indeed, if we plot the phylomorphospace for this group, we observe that there is a considerable amount of overlap between among most of the lineages, except for the family Ruxiaceae and the order Spherocarpales, an order which is included within Marcantiopsida. Although we don't have a clear mechanism to explain this pattern, it could well be that the appearance of new morphotypes is somehow related to environmental factors or requirements. Indeed, if we consider the life strategies of these lineages, we observe that most of these families that overlap in space are mesophytic and non-ephemeral, as compared to Ruxiaceae, which are xerophytic and ephemeral in the life cycle. So I think I'm going really fast, so I'm ending with some remarks. So first, integrating fossils into total evidence phylogenies allowed us to assess their systematic affinities. And this challenges the common view in the field of bryology, which argues that fossils, bryophyte fossils, are too fragmentary or poorly preserved for being assessed from a systematic viewpoint. Nevertheless, the placement of some fossils was sensitive to the analytical method, and for instance, Metriotalus, Palavicinites, and both species of Marcantites were sensitive to the weighting scheme and the optimality criteria. For this dataset, at least, including fossils and in the reduced dataset, including these fossils led to older divergence times, and these complex liverworts are inferred to have diverged in late Devonian instead of early Permian, which is quite significant. And it also allowed us to recover novel macroevolutionary patterns, and we could think that liverworts were already diverse by mid-Mesozoic. So with that, I'd like to thank all of my colleagues who contributed to this study in different manners, and institutions for giving access to collections, services, and funding. Thank you.